introduce Dr. Raquel Cowell. Dr. Cowell received her PhD from the Institute of Child Development at the University of Minnesota. There she worked in the Cognitive Development and Neuroimaging Lab, studying the role of context on decision-making in children and adolescents. Currently, she is a postdoctoral scholar through the Templeton Foundation Wisdom Grant, where she collaborates with Professors Jean Basidi and Ali Bartaksu, examining the neuroeconomics of decision-making. Their most recent collaboration will focus on the role of perspective-taking in monetary decisions within the fMRI environment. Today, Dr. Cowell will be speaking to us about the development of sharing and moral behavior in children and how this applies to our understanding of altruism and neuroeconomic models. Dr. Cowell.
Thank you for having me here today. I would really like this discussion to be somewhat informal in that if there's questions or if there's any points of clarification or things that you'd like to know more about, feel free to ask. I today will be talking to you about some of the things that we know about altruism in children and also ways that we've gone about studying pediatric populations and the challenges inherent in that. So my talk today will be divided into two portions. I'll start off by talking to you about altruism in children. This will include operationalizing what I mean by altruism and how that applies to developmental psychology. And I'll also provide you with a laboratory experiment example of how we typically measure this and some of the challenges inherent in that model and ways that we're trying to overcome those obstacles, as well as some recent findings that collaborators have come across. And I'll also like to talk to you today about brain development in children, what we currently know about brain development in children, as well as the methods that typically are used to study brain development in children. So to begin, I thought it'd be useful to kind of make sure we're all on the same page when one says altruism. So what is altruism? Well, an altruist is willing to reduce his own consumption in order to increase the consumption of others. That comes from Darius Becker, and I thought that this particular image really illustrates that example. I don't know about any of you, but I'm a really big fan of ice cream. And growing up with little sisters, I was known to growl if they tried to take my ice cream from me. And so to be altruistic would have been me being willing to give them some of my ice cream with the knowledge then that I would not be able to have whatever I gave them. And so that's kind of the basic working model of what altruism is and how it's currently defined with neuroeconomics, particularly with developmental work with kids. So what is altruism in children, and how do we study it with them? Well, in children, it's helping without receiving praise or concrete reward, and it comes with a potential cost. So in kids, this potential cost is a little bit more challenging to study than in adults. With adults, a lot of times it boils down to money. So you can often do paradigms with adults where you give them a certain amount to see how much they're willing to give. With kids, you have to find things that motivate children. And then that also can vary based on what population of children you're looking at and what particular age of children, because not all kids like the same thing, especially if you're going for different ages. So if you're going for someone that's really young, say someone that's 18 months old, what they find to be rewarding and what might be a concrete reward or something that they're giving up is going to be very different than, say, an 8-year-old, right? And so those are some of the challenges that developmentalists have to come across, is how do we translate things across these ages? And what are some common ways that we can do that? There's been some work being done on, is altruism, this willingness to give at the cost to oneself, is this unique to humans? Or is this something that might be shared with other non-human animals? And that's still definitely up for debate. But I wanted to share with you a video that's taken from a recent study by Felix Wernicken and Thomas Sell over at Harvard, where they looked at this in chimpanzees and children. And particularly, this is going to be a clip of a little child. Don't worry if you can't hear the audio too well, because they are, that's really much more important is the body language. So you're going to be seeing 4 different views come up on the screen. And I want you to pay attention to the female researcher that's sitting at the desk and the baby. And I'm going to have some questions to ask you guys after we do that. maybe he had to stop playing with his toy to go and get the pen to hand to the woman. Did the woman say thank you or anything to the child afterwards? Did she smile or anything like that? No, so that was the reward. That wasn't there. So the child still gave, even though there was nothing really gained from that. And so that's an idea of how you would measure that in children. Yes?
Was there any procedure in place to show that maybe would have expected a reward from the child?
That is a question, mainly because a lot of times kids are kind of, after a while, they learn from their parents, like certain behaviors are expected. In this particular one, they tried to be very careful in making sure that the child, there were several trials, and they wanted to make sure that the child wasn't expecting to receive the reward. And they were trying to match that because they'd done this in chimpanzees as well. They were trying to make sure that there wasn't this contingent learning going on. But that does bring up some challenges, particularly with young children, because you can't bring a child into the laboratory without their parents present. And so you get these other interesting pieces that you're not expecting. So there was some research that was done where they were wondering, okay, are children only helping because their parents are in the room? Are they only helping because their parents expect them to help? So what they tried to do is they tried to make sure that the parent wasn't in the room to see if they still engaged in spontaneous helping behavior. And they found that yes, children often still engage in helping behavior when their parents aren't around. But that is not necessarily always the case too. It's just in maybe I would say 33% of the sample that kids did that. A lot of times kids don't always like to share, generally. But that's still difficult to quantify. And you also get into other challenging pieces of when a child's in a room without their parents. So is that typical of what the child expects in their environment, particularly with kids that aren't maybe used to like daycare settings or that kind of thing? So there's definitely a lot of ways that this work can still be tailored and can be examined, but that's kind of a general model that's used to look at that in young children. There's also some work that's been done actually by economists here, by John List and Annie Samak, who's actually now at Wisconsin, who they looked at altruistic giving in 3 to 5-year-olds, and they looked at it slightly differently because there's this idea with economists that people give, and even if it's considered altruistic, there's some amount of reward involved. Just by virtue of giving, you get a certain amount of warm glow. And so researchers were like, well, if you still find some kind of warm glow, is that really altruistic? Is that really altruistic if there's not really a cost to yourself because you find it enjoyable? So I don't know if any of you are familiar with watching Friends. So there was a character on there, and her name was Phoebe. She was kind of this hippy-dippy kind of girl, and she was saying that there really was no such thing as a selfless act. And so her friends were trying to find ways of proving that that was not the case, and they came up with the example of a bee. The bee stings, and its selfless act is dying, but Phoebe's argument was that it's still looking cool in front of their friends. So there's this idea that maybe no act is completely altruistic, but there's ways that we still want to try to get at that, because over time, we're interested in altruistic acts that you see in children. Those carry over into adults. If as a child you exhibit helping behaviors, is that something that you'll be able to find later? And if you can help facilitate that at an earlier age, is that something that can help make us a more caring kind of population later on? Alright, so I wanted to show you just kind of a typical example to try to contextualize a little bit about how this research takes place. So I wanted to talk to you a little bit about one of the games that's often used. And as I'm presenting it, I want you just to kind of pay attention to how it's done, but also think about how this might be a little bit challenging to take and to put into either an fMRI or an EEG environment to study like the neural underpinnings of things. So this is the dictator game. You have a child come in the room and you say, here are some stickers. Choose the stickers that you would like. And you show them the stickers and you let them choose like 10 of them. And it's important that the kids get to choose those stickers, and there's some debate on that as well, because if the child's able to choose them, then you know that the child finds those particular stickers pleasing. So for example, if the child really loves panda bears and you only give them horse stickers, then it might not really be the same measure as if you gave the child stickers that they really cared about. So there's all these ways that you can kind of play around with that too. So here's some stickers, choose the ones that you would like. while I'm gone, will you put these stickers that you would like to share in this envelope? And then I'll come back and you'll keep the rest of the stickers that you didn't want to share. So then what that does is that allows the child to decide without anybody watching them how many stickers they want to share. So the experimenter either turns their back or leaves the room entirely and provides the child with an envelope so that they can put it in and it's a secret. No one else knows how many stickers. Assuming that the researchers can't do math, but that's a different story. So they put them in there, and children really do vary. In fact, this research is currently going on in my lab right now, and last week there was these two twin little boys that came in, and one of the twins shared half of their stickers, and the other one shared none of their stickers. And so this can really vary based on child. So some children share a lot of them, some don't share any, and that actually carries across in adults too.
Well, I know this would be an anecdote, but were they monozygotic or dizygotic?
I think they're monozygotic,
Oh, wow.
like that's why we were surprised because we were expecting, there to be more correlation between their behaviors, but no, one of them just did share. One of them in fact was kind of, I don't know if you can say that about kids, but he's kind of a jerk. But you find that, a lot of times people expect because they're kids that, all kids are sweet or all kids are nice, but it's not true. And a lot of times, aspects of a child that you can pick up on, you can carry over into later. And so that's part of why it's so interesting to study children. So that's one of the ways that this is done, and they've also done this with kids where they had an in-group and an out-group. And so the way that these in-groups and out-groups are chosen isn't always very clean. It's very difficult to create a paradigm with pre-established in and out groups. And so it's hard to really get at that. So some of the ways that researchers try to do that is by out-groups that already exist, like gender. So at younger ages, there really are clear distinctions between who plays with who in the playground. So girls play with girls, boys play with boys, and that doesn't really switch over until around middle school. And so that's a way that researchers try to get at this in-group, out-group. Sometimes they'll do things like assign one group a certain color shirt and another group, but you have to allow for time before you can really reliably test that they acknowledge that they're in different groups. And so those are some of the challenges that really wanting to make sure you're getting at that. But some of the work is, okay, how many of these, let's say that they had the 10 stickers, how many of those times did they share? And this is at different age groups. You have 3 to 4-year-olds, 5 to 6-year-olds, and 7 to 8-year-olds. And the black solid line is out group, and the gray dash line is in group. Is there anything in particular that you guys notice that is different between the 3 to 4-year-olds and the 7 to 8-year-olds?
In one group.
Yeah, so if you notice which line is higher for the 3 to 4-year-olds? The out group, right? And then it switches over to 7 to 8-year-olds, and there's really not much of a difference between 5 and 6-year-olds. So one of the ideas is that maybe at 3 to 4 it's kind of an artifact, maybe because they're both pretty low, they're not sharing that much in general. And then if you notice, sharing behaviors increase over time, but only for the 7 to 8-year-olds and only for an in-group. And in fact, it decreases from before. Yes?
Are the error bars just so tiny that we can't see them?
The error bars were conveniently not included in this particular research paper, so I didn't feel like digging through all the stats for you guys, I'm sorry. But yeah, the error bars are, for kids, they tend to be rather large, so I think that they kind of left those out to try to get their argument across. Not that that ever happens, but yeah. So, but the idea is that there are some differences between the ages over time, and the thing that I thought was interesting was that at 5 and 6 years old, there really isn't much of a difference between the groups. And that could be because they don't really see groups yet, or it could be that they don't necessarily care. And I think that as you get older, those kinds of things start to come out more. And so you start to see these differences that maybe younger kids are just kind of like, sometimes they feel like sharing and sometimes they don't feel like sharing, so you can't really pick up on a clear pattern, whereas as they get older, you start to pick that up more. All right, so here's a question. Why do we use, what useful pieces is neuroscience providing us in studying these kinds of things, in studying altruism, and studying moral behaviors, pro-social behaviors in children? And if you have a really strong answer, I would love for you to tell me, because it's still not always clear what exactly neuroscience buys us, apart from letting us have one more level of insight into what's going on in human development, right? But that's just an additional level that we're having to try to get at what's going on to remember that we're still at a stage in research where we're trying to compile all this evidence and we're trying to use it to create a clear picture of what's going on with humans, but there's still a lot of limitations, and particularly when you're looking at pediatric populations. And I would like to kind of have you walk away with this understanding of it isn't always easy, but it's definitely worth it, and we do find some really cool things out that we wouldn't have known otherwise. But to know that just because someone says it's in the brain doesn't mean you don't need to question it. So there's some convergent evidence going on to suggest that there's something that is happening in younger children that can carry across time into adulthood. Neuroscience can provide us with a greater scope in hypothesis testing. So instead of just having one measure, so let's say we have a behavioral measure, and the child shares half the time, we can get a sense of maybe the child shares, but maybe there's some kind of internal struggle. Maybe two children engage in the same behavior, but the underlying process leading up to that behavior is different, and you can pick up on those kinds of things. And so those are some of the pieces that are useful that you don't get without a neuroscience perspective. And so that's something that I always think is interesting too. You can get to the same endpoint behaviorally, but you might not have any clue about all the intricate things that are going on leading up to that point. And neuroscience can often help you do that. So how do we study brain development in children? Well, I thought first it would be handy to just kind of give you a basic, fundamental foundation of what we know so far about brain development in children. So this is an example of neuronal growth across the lifespan and synaptogenesis happening. And so you see these neurons in the newborn and across time. And as you notice, it starts to become, there's this greater connectivity between neurons. And that's important. And something I wanted to point out was notice at 2 years old, how there almost seems to be a greater amount of connections going on than even in adults. And that's because children start off with an overabundance of connections, and it's the experiences that they go through, and it's the all the things that come out in their life that show that this is relevant to their environment, this is relevant to their biology, that you start to see pruning away of certain connections. And so that's kind of an example of that. This is very much like a general idea of what's going on. A little bit too about what we know about the time course of critical events that are important in human brain morphometry. So there's a lot of work that we've borrowed from non-human animals. Sometimes it's challenging to get really into it in humans, particularly in living ones, because of the limitations we have in our methodologies. But there's a lot going on, and if you notice, most of what's happening is before birth. So that's something to really point out in general, that a lot of the big changes that are happening in the brain happen before a child ever comes out of the womb. And that leads us with starting more and more to realize how important that time of life is for the child. And so there's this period of time where we are particularly interested in. Researchers are very interested in this period because it really allows us to get at the importance of the environment and playing in with biology and trying to help us predict what happens in adulthood. Because by the time people get to adulthood, a lot of changes are kind of solidified. And it's the idea of how these changes have taken place over time and why is really what we're interested in. But it's also a very challenging time to look at as well. So we know that there's synaptogenesis going on. We know that there's some pruning of important neuronal connections. But a lot of what's to be determined is which of those connections become strengthened. So through myelination, which is something that occurs all the way through, it's up for debate, but usually your mid-20s, your brain is still undergoing myelination, particularly your frontal cortex parts of your brain that are really important for reasoning and inhibitory control and that kind of thing. But a lot of what's happening in the younger age group is just overall input in which neurons are forming connections. So some interesting things is that by age 2, the human brain is at 80% of its adult weight, and by age 5, the brain is approximately 90% of its adult size. So a lot of major life events can happen before the age of 5 that can actually impact a child forever in a lot of sense. So one of the populations that I had studied at the University of Minnesota were children that had come from post-institutionalized backgrounds. So children that were adopted from orphanages and internationally adopted to homes in Minnesota. And looking at the orphanage care that they experienced and what age they were adopted for everything up to how they share with others, how they relate to others, and how well they are able to pay attention in doing overall classroom settings. So this particular phrase, I think, really sums up where we are right now in neuroscience. I think that it's important to remember this, that the brain is wrapped in a resilient membrane that's immersed in a protected moat of fluid, and it's completely encased in bone. This protects the brain from falls or attacks from predators, but it also creates a challenge for those interested in investigating the organ responsible for our thinking and our behavior. So a lot of times to study typical and normal development in the brain, especially in children who are a very vulnerable population, there's a lot of challenges that we have to go through. So what are some of these challenges, particularly with kids? I'm talking about children. Well, one of them is fear. You get these little tiny preschoolers coming in, and all of a sudden they're surrounded by these strange adults in this very strange environment, and you want them to do these tasks like share stickers with me, please. And a lot of times they can be overwhelmed. And so the question with that is, okay, well, are we actually picking up on things that would happen outside of this laboratory environment? What are some ways that we as researchers can try to make this environment more comfortable for children so that we are confident that what we're observing are reliable things that happen outside of this particular context? Another thing that can happen with kids is boredom. Boredom is huge because have any of you volunteered to be in like a research study generally? Have any of you been bored in those research studies? A lot of times for us to get reliable data, we have to go through the same process over and over, trial after trial after trial, the same thing. So, for example, with EEG, if we wanted to do the dictator game, where we give them stickers and then we ask them how many stickers they want to share, to be able to get a reliable signal, we probably have to do in the hundreds of that. So could you imagine asking a 4-year-old child to do that 100 times in a row, sitting still in a room with strangers? So that's the challenge. That's the challenge we as researchers run into. Well, how do we make this something that we can reliably get from the child? And you can every once in a while get a really well-behaved kid that'll sit there, but that's not typical, particularly at certain ages. So one of the things is boredom or inattention. So I worked a lot with preteens where they're like 12 years old, 11, 12, 13, and you're just trying to explain the rules to them, and they're just looking over and they're not paying attention. And so getting them to not only listen to you give instructions, but then to sit still at the computer while you are hanging out in the background and actually trust that they're doing it and taking it seriously, that's challenging. Movement. So in fMRI work and in EEG work, you have to be able to stay really still. If you don't, the signal to noise ratio increases and there's a lot more noise in the signal. And when that happens, it's not quite clear if what you're getting is reliable and if it's actually from the biological system that you're trying to measure. The other thing is executive dysfunction. So especially in a lot of populations of interest, they usually have ADHD or other things that make it a little bit more challenging to study them in addition. So kids in general are kids, but then if you add anything else, it starts getting a little more complicated. Then you have some kids, especially, so one age group that's particularly challenging, we try to get them in to do EEG with them where we put the electrodes on their scalp to measure things, is 18 to 24-month-old children. And they don't like things on their head. If something is here, they've developed enough motor ability that they will go like this. And they will rip off that $1,000 piece of equipment off of their head because they don't like it. And you can try things like we gave them swim caps, sent home to the parents could practice with the child, and like all sorts of things, but they have this new power where they can use their arms and they can do things so that we can't put caps on them anymore. So between about 18 months to about 3 to 4 years of age, that's like an untouchable zone for doing any kind of EEG work because kids they're just not there. So that's a challenge. Also verbal abilities. So in the first video I showed you, that child was pre-verbal, right? So he could laugh and play, but getting a sense of, other kinds of paradigms that you might do with an adult. You can't really put an adult person in a room and have them play with a game and then have them reach over and pick up a pen for someone, and it's not really the same thing as studying helping behaviors in adults as you would with a child. So trying to make sure that these things extend across time is one of the challenges. And also parental interference. So sometimes parents really want to be involved and they really want their kid to do a good job and they really want the child to impress the researcher. And so the child has this external pressure to do well that maybe isn't reliable. Or sometimes if they're particularly young, the child will sit on the parent's lap while they're doing a task. And they pick up signals from the parent without meaning to. The parent will send signals to the child, rate of breathing or physical gestures that the child picks up on. So sometimes if the parent has to be in the room if it's a younger child, then we will blindfold the parent. So the parent will be sitting with the child blindfolded so that we know that they're not biasing the child's responses. And some of the other challenges involved with kids and adults as well is hair products and hair types. And not all hair types actually work well with the EEG system. So this summer I spent a lot of time over in Chicago Heights trying to do research with children who are coming from low-income, high-diversity backgrounds, trying to have them come in and do an EEG study where we're looking at inhibitory control. And these kids were adorable and really well behaved, but because of like if they had dreadlocks or if they had beads or anything like that, suddenly we couldn't put the electrodes on. And short of asking them to cut their hair, there's some children where it's not going to happen. And I was really hoping that we could do it because a lot of up until this point researchers just say I don't want to try. So we really tried and we really talked to parents ahead of time. We really worked with the researchers to make sure that they were well trained. And there's just some hair types that just aren't really compatible yet. And so until we find a way to overcome that particular challenge in our technology, there's some limitations in the questions that we can ask. So there's two ways often that we go about studying the brain in children. We study the morphological aspects, so the structure of the brain. So these are things like how big is this child's brain? What's the volume of different structures? To the function. So there's several ways that we do it. The two that I wanted to mention to you today are functional magnetic resonance imaging, often referred to as fMRI, and electroencephalography and event-related potentials. And so those are the ones. fMRI is the one where you have the tunnel, and the child goes into the tunnel, and I'll show you some images. And EEG and ERP are the ones where you have caps on that pick up electrical signals that are emitted by the brain. And so I won't get into the specific technical details, but if you have any questions about that, then I would love to send you to the right place or provide additional information. So in studying structure, a lot of what researchers like to look at is the morphometry. And so children that have typically developing in children from environments of early adversity, you can look to see if their structures are similar, particularly structures of interest. So some structures of interest for researchers are the hippocampi and the amygdala. Hippocampi is really important for memory and learning, the amygdala for like emotional processing and fear. And so looking to see if maybe those structures might be different in different populations of children based on experiences that they've had before. And so one of the ways we do that is we have them be in an MRI scanner. And this can work with almost any child that is sedated because they stay still. So they're in a scanner, they stay still. You can do this with newborns. So you can swaddle up a newborn and the mother can rock the newborn until the newborn falls asleep. And then you put the baby in the scanner and you can get a sense of their morphometry. This doesn't work for functional aspects. So if you actually want to know what they're doing while they're doing an activity, what their brain looks like, this doesn't work for pediatric populations until you get to about 7 to 8 years of age. So this is the scanner that we have here at the University of Chicago, over at the Brain Research Imaging Center. So this is the one that I've been using. And we've had kids as young as 7. They've claimed that they've gotten a 4-year-old in there, but I don't know how long that 4-year-old actually stays still for them. So that's up for debate. But this is the scanner. And you can imagine as a little child, because this scanner makes sounds. It grumbles. Depending upon the image it's trying to get, it sounds like a giant metal monster. And so you can imagine for a child how intimidating that environment might be. And so what we try to do, particularly with pediatric populations, is we use an MRI simulator. And so what this does is it allows the child to come in, and researchers will dress it up in different ways. So here at the Brain Research Imaging Center, we have it like it's in a space room, right? So there's planets hanging down, there's glow-in-the-dark want to get in the simulator. And so we had to go and have her play in the tunnel for a while. She really loved that. And then she made me get in the tunnel. And then when she felt comfortable with that, then we went to the simulator. And she had to have me do it first to make sure it was safe. And then she did it. And then she was okay with doing the actual scanner. But it took several stages before this little girl was ready to get into that giant machine. And so this is really helpful because when you're in there, what it actually allows you to do is you can even have a computer task, and you can have a laptop set up where it has a projector so the child can practice the task inside the tunnel at the same time. And that really is great because it allows you to practice both the task and get a sense of what it's like in that small space. So sometimes people think that they're going to be okay in the small space, and then you put them in there, and suddenly they get hot flashes or cold flashes, and they just can't be in there anymore. And a lot of times with populations where you're worried that they might get nervous, particularly children, you always want to give them an opportunity to see what it's like first. And so anybody doing pediatric neuroimaging, it's a really great investment to have, is getting a simulator where they can really get a sense of what it's going to be like. So these are pretty high-tech things. The lights turn on inside the tunnel, there's a fan, you can play the sound of the magnet. The bed actually goes in and out with a button that you press. And then you have a little mirror up on top of the head coil that allows you to actually have it so you see the image the same way you would while you're in the scanner. And so those are some really great features that are available to people that are wanting to do this with children. And there's also electrophysiology work. So both temporal, looking at time-related aspects, and spectral, looking at frequency of different bands of interest. And so there's temporal is where like the child would be doing a task in the scanner, and you're able, not the scanner, the EEG environment, and you're able to time lock it so that you can look at, particularly at this window of time, say between 200 and 400 milliseconds after a stimulus, after the stickers appear on the screen, you can look to see what their brain patterns are looking like. And you can look at different areas that we know are reliably correlated to other aspects of behavior. So here's some things you can also do with children where they don't actually have to do a task, where they don't have to do repeated trials. And this is really useful for being able to say, okay, there's certain patterns we pick up on with these particular frequency bands that are correlated to other behaviors, to other pro-social behaviors, to other sharing behaviors. And so this can become a reliable predictor, whereas you might not be able to have the child do that particular task yet in the EEG environment. You can have them do something else. So these are some waveforms of interest. Some that are particularly interesting to researchers looking at sharing and pro-social behaviors are the frontal alpha asymmetry that's to individual differences in emotion regulation, motivation, social behavior. And so children that have different kinds of patterns in this frequency band, also this is correlated with how willing they are to approach someone in a new environment or withdraw in a new environment, how able they are to deal with frustrations. So have you guys ever heard of a disappointing gift task? So this is something that developmental researchers have come up with to torture poor children. But you can have them, you have the child in the room with the researcher, and the researcher says, okay, I have this really cool gift, but you can't see me wrap it, so I need you to turn around. And so then the child turns around, and the researcher takes this wrapping paper, and they make all these sounds and all this, and then they wrap up this gift, and then they give it to the child, and they have the child unwrap it, and it's a paper clip. So, based on the child's creativity, you can, but some children, they'll get this, right? So they'll open the present, and they'll see this paper clip, and they don't want to disappoint the researcher, so they'll pretend that they like it. And other kids will start crying, and that kind of thing actually is a pretty good predictor of other behaviors later on. And so you can get at a child's temperament or their emotional reactivity based on these kind of silly but actually pretty predictive kinds of tasks. Have you guys ever, this reminds you, completely relevant, but Jimmy Kimmel, where they have the parents tell the kids they ate their Halloween candy? I like that, but that's not, I guess the right hierarchy, so they do this task instead. So, depending upon which part of the brain is more or less active, you can kind of get a sense of if the child is more approach-oriented and have more positive, or if a child is more likely to withdraw in an environment. And this is something that researchers again and again and again, and yet it still provides predictive power. So it's one of those things that hasn't seemed to go away yet. This is not the same thing as saying that your left and right brain know. That's something else. This is frequency bands and activation. Yes?
Is it, so in adults, my lab finds out it's the reverse in lefties. Is that true for kids as well?
Probably. If we had enough lefty kids come in, yeah. I don't know about all labs, but I know in our lab when we had children come in and we test for handedness, if they were ambidextrous or left-handed, we would just immediately ignore that and just have them do it with their right hand, because things get a lot more complicated when you start to try to factor in handedness. But yeah, I'm sure that there would be some flipping on that. All right, so here is an example of how we would get at those frequency bands. You would have the child just kind of sit, relative rest. It's about 5 minutes. You get 5 1-minute clips with breaks in between. And then you can look to see if there's differences in left and right. And so you would have the child sit in the space paradigm. They're an astronaut. They're going into space, and so they watch a space movie. It's not available. So you get the gist. You're watching, you're in space, and the stars are coming at you and that kind of thing. And the kid just sits there for 5 minutes. And they try different ways. Basically, all you want is the child just to be paying attention and not really engaging in any kind of active activity. So just kind of looking at something is a great way of getting at that. They've tried to do other ways. So in some labs, they have like bingo balls where you put them in the cage and you roll the cage, and the kids will just look at that for a while. But this goes more with the astronaut theme, so it's more effective. So I just wanted to put in a plug for the child nurse suite here at the University of Chicago. In this lab, we're currently trying to do multi-levels of analysis to the study of social development. So there's a lot of things that have not yet been done in the field that we're trying to approach. These involve behavioral assessments, so that's typically the approach being used, as well as EEG, ERP, psychophysiology, so a child's heart rate, skin conductance, genetic measures, and social environment. So trying to take into these other pieces like a child's family structure, how that might be important. And then these are some cute pictures that have been done well in our lab. So this is from this summer, and this is the new system that's actually really handy for kids because you just put it right on, and then you can, while the kid's playing on the iPad, you can make sure that all the electrodes are ready, and you can get scissor under, and the kids like it because they're really cool colors. And the colors actually correlate with whether the electrodes are ready or not. So if they're green, you're good. If they're yellow or red, you still need to make sure that there's enough gel in those electrodes. And this has actually been a little bit more user-friendly for different hair types too, which is really nice. And I think there's some researchers, including Austin. So this is some of the work that's being done here. So do you have any questions at all? I know that we're about at time, so if anyone's tired, I understand. But, yeah.
Has your lab or any other researchers observed any noticeable differences in altruism or prosocial behavior in children of varying cultural and religious backgrounds?
That's a great question, and that work has yet to be undertaken. We're actually currently doing that. And so Professor John Steady recently received a large grant, and he's doing cross-cultural work right now with people from, currently the countries involved, we have Turkey, South Africa, China, Israel, Colombia, and Kyrgyzstan, and I think Canada. And so they're trying to see if these kinds of shared behaviors are similar or different across cultures. I know there's been some work done on kids from varying backgrounds in terms of socioeconomic status, and the argument is that in those ones they've shown that high socioeconomic status kids share more. Being someone that came from a low socioeconomic status, I would like to say I disagree, but I don't know for sure. So I think that there's certain things where kids from higher SES also have parents that are much more involved in research and are more familiar and comfortable with those environments. So there's still some work that should be done there too.